Thank you.
Okay. Thanks for coming, everybody. We are happy to have Will Oestreich here. Will's currently at the Monterey Bay Aquarium Research Institute, and before that, with a PhD at the Hopkins Marine Lab, Stanford. Undergrad. And as far as I know, he spends like all day listening to creatures in the ocean.
More or less.
Singing fish and things.
Yeah. Yeah, thanks, Peter, for the introduction. Are you all hearing me okay in the back? Cool.
On Zoom, it looks like they've got this screen instead of their main screen.
Oh,
It was fine before, I know.
try this again.
Changed one thing and now messed up again.
Is that working now?
Yeah, it's still
Huh. Oh, swap. Okay.
Then it'll put his near display. Yeah. Still.
That's funny. We had it. Oh, sorry. Yep.
At the top. Swap. No. That's just swap though.
But you want to change which thing you can share in Zoom. So stop sharing. And now share again.
Yep.
And, yeah.
There we go.
Okay, this one here. Yep. Is that better?
Doesn't show. Doesn't start sharing.
Where did Zoom go?
Yeah. So, oh yeah. So if I go to,
So you'll want to have your presentation like sort of already,
yeah, like playing from the,
yeah,
yeah.
so we'll just make out sharing it and then we'll go back to Zoom.
Yep, that was it.
Okay.
Yep. This is Zoom.
Okay. So you already shared it.
Right. That's what I was trying for. Here, I can that one.
Okay, lovely, yeah. And then
Okay. Is it showing up on Zoom?
And go up to the top.
Oh, and then resume.
Can you go up and hit resume share on that screen?
Oh God.
Like,
Share. Not like. That's not other. That's not.
Yeah, look what you go.
So sad.
We have to get the floating display back on. Okay, should we just?
Yeah, I think we just stop
so many times.
The top one, is that what we did?
I was in the Microsoft point. Yep.
Okay.
Exactly.
Screen sharing.
Yep.
Okay, so let's go to presenter. Still on Zoom, but maybe that's okay.
Yeah. What if I just go like that? Is that better on Zoom? Is that better on Zoom? No, or I just. Oh, okay. Cool. Okay. All right. All right. Thanks everyone for the patience and sorry about that. And thanks everyone for having me and for the kind introduction, Peter. Yeah, it's really exciting to be here and really lovely to meet some of you all so far. Looking forward to meeting you all over the rest of the day and a little bit next week. Yeah, but without further ado, I'll go ahead and jump into some research that I've been thinking about with collaborators, studying the collective behavior of organisms which inhabit oceanic ecosystems. And when thinking about the behavior of oceanic animals, I like to start with these krill, because I think they're a great representative of what life in the open ocean is like. So these are among the most abundant animals on the planet, both in terms of the number of individuals and the biomass. And despite being extremely abundant in the ocean ecosystem, they're actually absent from most of this ecosystem. So their distribution is extremely patchy. As we can see in this image on the left, this is an image of a survey for small schooling fish, similar to krill in a lot of their life and behavior. But we can see for a lot of these forage organisms that really form the middle of marine food webs, they're extremely abundant, but they're extremely patchy in their distribution across nested spatial scales, everything from within the swarm itself to the scale of oceanographic features and ocean basins. So not only are these animals extremely patchy in space, but their location of these patches are extremely dynamic in time. And this is because, of course, the ocean is constantly moving. So here we can see an animation of primary productivity in the surface ocean. We can see that as the ocean currents are moving, biological hotspots are being moved in space and time too. And this creates a really interesting behavioral dynamic for these organisms, which live really right at the plankton-necton edge. So these animals have some planktonic characteristics where they're being forced by the physical movement of the ocean in space and time, but they also have behavioral agency and can make active behavioral choices despite being subject to these physical oceanographic forces. Of course, another key element we have to consider when we're in the open ocean is this enormous third vertical dimension, which really introduces this incredible gradient of light in the ecosystem where there's really no light available throughout most of this volume that we're studying. And this, of course, can select for really different sensory modalities in terms of how animals are communicating, how they're searching for and finding prey, how they're evading predators, so on and so forth. So as a behavioral ecologist who's really interested in this interface between animal behavior and biophysical ecosystem dynamics, I'm drawn to comparing what's happening for life in the open ocean to what's happening in these more heavily studied ecosystems on land. And really, we have, as I've been talking about, these really big differences in terms of ecosystem scale and structure, the spatiotemporal patchiness of resources in the land or seascape, the sensory modalities and communication ranges that this selects for. And so if we think about these differences between the ocean and land in terms of the spatiotemporal ecosystem dynamics and the perception, sensing, and communication systems of animals inhabiting these systems, there's been a really big growth in behavioral ecology in recent years, showing that these are two factors which are really strong mediators of social and collective behavioral processes for animal groups. So as an example of this, we've had a big proliferation of theoretical papers trying to understand how spatiotemporal ecosystem dynamics select for different social information use patterns in animal groups, how the perceptual ranges and strategies for information gathering and group sizes are dependent on spatiotemporal ecosystem dynamics. And then, of course, how these sensing and communication dynamics drive collective behavioral processes in different ways in ecosystems that exhibit different spatiotemporal ecosystem dynamics. Just to highlight one piece of work here that I got to work on with an incredible undergraduate researcher, Kanoi, who's now doing a master's degree working on some other questions I'll talk about a bit later. I won't go into the details of this review paper that we wrote about collective animal migrations, but I do want to highlight one thing, that as a part of this review, we did a systematic literature review of cases where social dynamics play a major role in animals' migration timing decisions. And you'll see there's a great diversity of taxa that this idea has been studied in, but one of the things we really noticed is that all but one of the cases we were able to find was happening outside of the open or deep ocean. So really only one case in that massive volume for life on Earth. And so if we return back to the differences in terms of how we think about these terrestrial and oceanic ecosystems, another key difference here is just the degree to which we have detailed observations of individual and group level behavior to explore these connections. I don't have to remind anyone here, but just to really hammer the point home, We, of course, are living on a planet that's dominated by oceanic ecosystems. These deep water, open ocean ecosystems by volume are the vast majority of the habitable space on our planet. And we still know very, very little about what collective behavioral and social behavioral dynamics look like in this system. And importantly, we really don't know how behavior feeds back into ecosystem function in these systems, which play a big role in mediating our global climate system, for example. Of course, these are not just really dynamic and enormous ecosystems, but they're also ecosystems that are rapidly changing, like all other ecosystems on Earth. So, for example, we're seeing a variety of climate change impacts in these ecosystems, such as massive marine heat waves. And we're also seeing more direct anthropogenic impacts as our oceans become more industrialized, in part to address the climate crisis itself, for example, with the construction of offshore renewables. So in my research program, I really am interested in thinking about animal behavior in these dynamic and changing oceanic ecosystems, and really from a variety of perspectives. So first, I like to think about biophysical ecosystem dynamics, how that drives prey distribution and behavior, and how that interacts with predator behavior and distribution. And then sort of the enveloping umbrellas that I really like to think about these dynamics under are how these processes interact to drive animals' collective behavior in groups, and how this enables animals to either make adaptive or maladaptive behavioral decisions under anthropogenic change, and how these animals interact more directly with anthropogenic influences. So to get into a few more specific research threads under this broad umbrella of what I've been thinking about a lot about over the last few years and thinking about moving forward, I'm going to start with a series of stories about blue whale predators and their krill prey, thinking about the collective behavior in this predator-prey system under ecosystem variation and change. So just to orient everyone to the system that we'll be studying this in, here we're looking at a beautiful satellite image of the west coast of North America. Most of my work studying these animals has taken place in Monterey Bay, so really right in the heart of this California current large marine ecosystem and its incredible biological productivity. Notably, the Monterey Bay is also bisected by this deep submarine canyon, roughly comparable in size to the Grand Canyon, which provides really incredible habitat for abundant krill swarms. And this is also right at the heart of blue whales' summer and fall foraging grounds. This is the Northeast Pacific blue whale population is an endangered population of roughly 2,000 individuals that spends somewhere between 4 and 6 months every summer and fall feeding off of the west coast of North America. And they're feeding off the west coast of North America because of the incredible biophysical processes we have happening off of this coast in the spring and summer, leading to a proliferation of life. So this is seasonal upwelling, the process by which northwesterly surface winds interact with the rotation of the Earth to drive surface water offshore. That surface water is then replaced by this deep, cool, nutrient-rich water that is injected into the sunlit surface layer, leading to an explosion of life that propagates across trophic levels as the season progresses. Notably, this is what drives krill prey abundance in this system. This is a heavily studied relationship here, as this is really a keystone prey species in this system. So seasonal upwelling is not only driving the availability of this key forage species in the ecosystem, but it also varies in its timing and intensity. So here we can look at this sort of phenological plot of cumulative nitrate flux as a result of upwelling in this system. And you can see that there's an average annual cycle where upwelling intensity is peaking sometime in the late spring, early summer. But there's a lot of variability in terms of the timing of specific upwelling events and the degree and intensity of this upwelling process. So given this incredible interannual variation we see in the system, a question that I've been really interested in is as blue whales are making this decision of when to depart their summer or fall foraging grounds and head south to their breeding grounds off the west coast of Central America, how do they possibly make this decision under such interannual variability and uncertainty? And I'll just note that this is a really critical decision in the lives of these animals. They're going to feed very little, if at all, for the 5 or 6 months that they'll be off of the foraging grounds, where they'll be birthing and nursing their young. So this decision of when to give up on the foraging season and call it quits and live off of those accumulated fat reserves is really a critical one in the lives of animals in this population. So the central question here was trying to understand how blue whales decide when to forage and when to migrate. And this is work that I've worked on with a number of collaborators over a number of years now with funding sources listed here, which have provided incredible support for this work. The main tool that I've been using to study these questions is long-term passive acoustic monitoring. So this is a program that I've been affiliated with and working on with researchers at the Monterey Bay Aquarium Research Institute for a number of years now. This system is on a cabled seafloor observatory. You can see the location of that observatory in outer Monterey Bay with the white circle there, connected back to our lab by this roughly 50-kilometer long cable. And this hydrophone gets a continuous power supply and data feedback to shore through this cable. And it's been recording more or less continuously since July of 2015 now. This has been an exciting way to share data, analysis tools, ecological insights with other researchers and the public. And it's also given us incredibly detailed information about the lives of blue whales moving throughout this system. We've recorded blue whale songs for more than 1,000 days since we've started this recording over what we estimate based on acoustic propagation modeling to be roughly a 10,000 square kilometer area of detection for blue whales from this single instrument. So I mentioned blue whale song. What am I referring to when we talk about song? So this is a visual representation of blue whale song. You can see frequency or the pitch of the sound on the vertical axis here through time with the colors showing the intensity of sounds at a given frequency and time. And we'll see if this will play for us here. I'm not sure that it's going to, but given that it won't play, I'll just have to leave you to imagine what this lovely low frequency sound might sound like. But you'll also notice that it's very consistent and stereotyped in that these units, these A, B, and C units that this animal is producing are at very specific stereotyped frequencies. And we take advantage of that in the red band shown here to quantify the intensity of blue whale song in the ecosystem-wide soundscape at any given time, basically by comparing that target frequency band and energy intensity in that band to nearby background frequency bands that are not affected by blue whale song. And this gives us what we refer to as a call index, really just the signal to noise ratio in those target and background bands. It's really a proxy for population-wide song intensity from this single recording instrument. And so I mentioned that 10,000 square kilometer detection range that's shown here. The colors here are representing the area over which we can record songs from that single instrument in outer Monterey Bay. And what I'm about to show is an average annual cycle of what we record in blue whale song throughout the year, many years of recording condensed down into one average annual cycle. So first we can look at really song intensity, that call index I described. But we can also look at the time of day that these songs are produced. So in this plot here, we're showing the diel patterns of songs throughout the seasonal cycle with values above the dashed line at 1 indicating more song during the nighttime and values below indicating more song during the daytime. And to my eye, there's really two clear parts of this seasonal cycle here. There's this first part where the song intensity is ramping up throughout the summer and fall, and we're hearing the majority of song during this time during the nighttime. And then there's this overlapping period bleeding into the early winter months where song is dissipating, and we're starting to hear a more even distribution of song throughout day and nighttime hours. And I remember the first time I looked at this plot thinking there must be some other behavior that's driving these really clear seasonal and diel patterns that are linked in blue whale songs. And so to investigate what might be driving these population level patterns in song, we turn to individual level behavioral context for song. So these are biologging devices developed in partnership with a company called Customized Animal Tracking Solutions. They're attached to the backs of these animals using suction cups, and they carry a variety of instruments that allow us to recreate these animals' movements in 3 dimensions, also to record video and audio from their backs, as well as a variety of inertial data streams from inertial units similar to what you would find in your smartphone about the orientation and movement of your device. And so we place these devices on the backs of animals. This is work that I did during my time at Stanford University with Jeremy Goldbogen's lab. And I'll just give a quick shout out here to my colleague and collaborator, James Fallbush, for many, many hours that we spent together, the two of us on a small boat like this, searching for whales, deploying tags on whales, and perhaps the greatest amount of time searching the ocean, trying to find these tags after they pop off of the whales, so we can recover that valuable data. So many days at sea in this boat that's much smaller than the animals that were studying themselves. We'll see if this video will play, just to give you a sense of the sorts of amazing sights we can see from the backs of these animals. Yeah, this one's going to go. Oh, it's getting hung up a little bit here. I'll just fast forward to some critical freeze frames here. Here we're seeing a blue whale feeding on a swarm of krill as recorded from the back of another blue whale. And so you can see a couple of key freeze frames here as this whale in the front now has engulfed a bunch of krill prey and is filtering it out. Over the next minute or so, this animal will be just gliding, filtering water out as you can see that inflated pouch on its belly running all the way from the tip of its rostrum all the way down to its navel. That's really how these animals make a living, accumulating forage from these densely packed krill swarms. To get into data a little bit more that comes off of these tags, it really gives us what we sometimes refer to as a daily diary into the lives of what these animals are mostly doing out of sight of us, well below the ocean's surface. So here you can see one individual tag deployment here, a day in the life of a blue whale in Monterey Bay. And you can see the black line showing the dives of this animal throughout the day. Those blue dots indicating every one of those feeding lunges like we saw in the video that we know from signatures in the accelerometer data. You can also see the song calls that this individual is producing, also recorded not just from the hydrophone that's on the tag, but also from the accelerometer on the tag as the animal's body vibrates, allowing us to discern calls that were coming from the tagged animal versus calls coming from other individuals recorded nearby. This is just an example from one individual here. But as you see, the day-night patterns that you can see up above of more feeding during the daytime and more singing during the nighttime, they hold true over many individuals over several years. So again, we're seeing most of this feeding happening at depth during the day and most of the singing happening at night. And this, to me, I remember when compiling this data and seeing such consistent patterns over many individuals and several years of this daytime foraging preference, it made me really start to think back to those population level metrics of like, okay, this is why blue whales are singing at night now, right? They're feeding intensively during the daytime and putting all of their energy and effort into that intensive diving and surfacing and breathing and lunge feeding that you see during the daytime hours. As we think about other behaviors that these animals are engaged in, I mentioned their migration to the breeding grounds. Sometimes we're also able to get these tags to stay on for more than a day. Here we're showing a month-long tag deployment. You can see the inset map here of the track that this animal swam over the course of this month. You can see the latitude of the animal here in the line on the main plot. And this dashed line is showing the foraging period. And as this animal's latitude decreases, this post-foraging southward migration to the breeding grounds. And we're able to take every feeding and singing event from this tag deployment and overlay it on this migration. So here you can see the circles indicating the number of feeding events per day, the triangles indicating the amount of singing every day. And you see that this animal has stopped foraging as it starts the southward migration, but it continues to sing throughout the migratory route. The other thing we can do is we can overlay the time of day that these behaviors are occurring. So you see here, the reds indicating a daytime bias, that red circles in the foraging period, again, indicating that daytime foraging, and these blue triangles during the foraging period indicating more song being produced during the nighttime. You'll notice that on the migration, we start to get a mix of blues and reds and whites. So this is really, we're hearing a mix of song spread roughly evenly over the day and the night. This is just one individual example. Again, we found this pattern to be consistent across multiple individuals. And so if we turn back to our population level acoustic metrics from the passive acoustic monitoring, what we realize here is we're actually hearing this population transition from the foraging state to the southward migration state. What we really now have is a population level acoustic signature of these animals' migrations. So a natural follow-up question for me as someone who's interested in how animals behave in relation to biophysical ecosystem dynamics was whether there's any interannual flexibility in the timing of this population level migration, given the interannual variability in the phenology of upwelling, which drives the abundance of their krill prey. I won't go into a lot of detail in this section, but I will just say first, we see a really wide variety of migration timings as we break this down, not now as an average annual cycle, but looking now at individual years of migration across this acoustic recording period. First, we can see a year like 2015, where these animals were migrating in the summer months, a year like 2020, where they're migrating as late as the winter months, and even years like 2017, where we're seeing multiple waves of migration, again, indicated by that decrease in the night-to-day ratio of their song at the population level. So yes, we see incredible interannual flexibility in the timing of this population's migration year to year. Okay, so are these variations in migration timing linked to the upwelling phenology? Again, I won't spend a ton of time on this section because there's a lot of oceanography involved here that I'm happy to talk about more later, but just remember that that upwelling process drives the availability of their krill prey. And we found that these animals were actually migrating later in years that had an earlier onset, a later peak, and a greater total accumulation of this upwelling that drives biological productivity. So these animals appear to be responding really flexibly and really dramatically to interannual variation in forage availability. And just to put this back into the context of their annual cycle, what we've shown here is that when they're leaving the foraging habitat, they have interannual flexibility that matches the phenology of forage availability. And to me, this indicates that they're using some sort of individual or social sensing of the ecosystem in real time to make this dynamic decision. Interestingly, this is the exact opposite of what my colleague Brianna Abrams showed a couple of years ago using tag data, in showing that when the animals are migrating in the other direction, they're extremely consistent year to year in terms of when they show up at different foraging hotspots. And to me, this is really makes some sense from a information availability point of view. When these animals are in the foraging habitat, they have information about what's happening in the foraging habitat, right? They have the luxury of being able to make a more flexible decision. However, when they're arriving on the foraging grounds, they're coming from a totally different oceanographic system. They don't really have any clear indicators of what forage is going to be like in a given year. And so they really hedge their bets. They play the long-term averages and show up in alignment with the long-term average phenology rather than the contemporaneous year phenology. One other thing I'll note here is that I said individual and or social cues. Well, which is it and how much of each? How are these animals actually making a decision of when to migrate when their foraging habitat is this incredibly vast and dynamic system? How do they accurately assess the availability of krill in this ecosystem? And this is work that I've been working on now to follow up these studies with my colleagues, Stephanie Dodson, who's in the Department of Mathematics at Colby College, and with Brianna Abrams, who I already mentioned in the Department of Biology just up the road in Washington. And we've been parameterizing a series of individual-based models using these detailed tag and passive acoustic recording observations of individual and population level behavior to test whether individual and or social information might allow animals to make more flexible and optimal migratory decisions in this system. So as an example of this here, you can see on the vertical axis, the timing of this population migration for an individual-based model where the animals only use their personally sensed information about krill availability in the environment versus a population that we've modeled where you allow them to eavesdrop on one another's behavioral state, listening for that acoustic signature of migration and hearing, even if I'm not finding food, everyone else is still feeding, so maybe I should keep at it. That's what's being modeled in this personal and social individual-based model. And if we compare that to the empirical population level metrics we've recorded from the hydrophone, we see that we get a much better match between our simulations and the empirical data when we allowed the animals to eavesdrop on one another's behavioral state. If we want to look at this at an individual level, we can look at from individual tags when animals started the southward migration. And again, we get a much better fit to the empirical data when we model this population in a way that allows them to eavesdrop on one another and use social information. Final note here is just that when we allow these animals to use social information, they acquire much more krill during the foraging season than they would otherwise if they were only searching for forage totally independent of other individuals in the population. So I just covered a whole lot about blue whales and krill here. So just to really recap and sort of center us again on what did we actually find here? First, there was a study where we showed that there is a population level acoustic signature of migration for blue whales in the Northeast Pacific that's indicated by the time of day that they're singing. Next, we were able to show that interannually, the timing of this population level migration closely tracks interannual variation in ecosystem phenology. And finally, by integrating these empirical observations with simulations, we think we're showing that these animals couldn't possibly be this efficient and optimal in their foraging or migratory decisions without eavesdropping on the behavioral state of one another over very, very large spatial distances. Okay, so that wraps up the first section of my talk, thinking about these collective behavior under ecosystem variation. Now I'm going to jump into some work that's a little bit more recent as I've been thinking about how to integrate tools in a similar way, these individual population level tools and simulation approaches to explore the collective behavior of even more cryptic animals that inhabit the truly open and deep ocean. So just as a reminder, because I can't help myself again, we really are living on this planet that's dominated by this habitat that we're really having a hard time observing. So one option that we have here is, I mentioned we did all of this work from a single seafloor hydrophone. We can also put an array of hydrophones out across an ecosystem, right, to understand some of these behaviors, perhaps further offshore, over greater spatial scales, et cetera. And this is exactly what I've been doing with the network of collaborators throughout the Northeast Pacific. We've been maintaining and monitoring a network of passive acoustic monitoring systems on the seafloor that have provided fodder for, I think, really exciting student projects. So for example, Emma Pearson was an undergraduate intern that I worked with a few years ago. She's now working on a PhD just up the road at Oregon State University. And she had a really nice paper using this distributed acoustic sensing network to explore spatial patterns of vocal and migratory behavior in blue and fin whales. Natalie Cross is another undergraduate student I've been working with to explore how these animals are responding behaviorally to marine heat waves, again, using this acoustic sensing network. But that really only gets us so far, just observing the behavior of large predators from these acoustic monitoring systems. If we want to really understand the process underlying these patterns, what I think we need is really detailed, persistent, and co-located measurements of the biophysical ecosystem dynamics, the prey distribution and behavior, and the predator distribution and behavior. And this has really been the focus of my current postdoctoral work at the Monterey Bay Aquarium Research Institute, where with collaborators, again, with from a grant from the Packard Foundation, we've been implementing a pelagic predator-prey behavioral observatory. So what do I mean by that? You can look at it in sort of a cartoon way here. Again, zooming in on Monterey Bay. Here's zooming in on really a specific part of Monterey Bay where we have this interesting mix of gentle slope into the abyssal plain and also these really steep shelf incising canyons. You can see the vertical cross-section here as predators and prey move around and are influenced by the physical movement of water. And we've been implementing a variety of integrated sensors to explore this system more holistically. So the first I already mentioned are these animal-borne biologgers we've been deploying. One that I haven't talked about as much yet, but which is really critical to this sort of work is, again, to understand the distribution and behavior of prey in the system. So we've been using an autonomous surface vehicle called a wave glider. This instrument makes use of the wave energy in the ocean to propel itself and uses its battery power to really power the payload it's carrying, which in this case are multi-frequency echo sounder system that allows us to really map where prey are in the environment and to discern what type of prey it is from differential frequency responses of the acoustic energy bouncing off their body and being recorded back up at the acoustic instrument on this wave glider. We also use a series of subsurface moorings. These carry things like hydrophones, but also a variety of physical oceanographic sensors. For example, the beams here, upward facing, represent an acoustic Doppler current profiler. This is a physical oceanographic instrument that allows us to map the direction and strength of currents throughout the water column to really understand how that's influencing the behavior and distribution of predators and prey in the environment. And the last one I'll mention here is that from that cabled seafloor observatory, we have the long-term passive acoustic monitoring system, but we've also implemented with collaborators a directional hydrophone. So this is an acoustic vector sensor. Typically, a microphone or hydrophone is measuring a pressure wave to record sound. Here, this instrument is measuring a different manifestation of the sound, the actual fine-scale particle motion, and it's using that to reconstruct a vector of where that sound was arriving from. So essentially, this is a hydrophone that points to where the sound it recorded came from. And that's been a really valuable tool for us in thinking about how to study social dynamics in the movement of groups of animals that can potentially communicate over really large ocean areas using low-frequency sound. Just to zoom in on a couple of different things we've been exploring using this system. Again, blue whales and krill. Clearly, I like them as a study system. We've been exploring really the predator-prey interactions here, not just at the seasonal scale of that foraging and migratory decision, but what drives the dense ephemeral krill swarms on which blue whales selectively feed, given that really energy-intensive sludge feeding style. They need to find these really dense krill swarms in order to make opening their mouth worthwhile from an energetic standpoint. And my colleagues at the Monterey Bay Aquarium Research Institute several years ago published a paper showing that these dense ephemeral krill swarms are much more likely to be found in plumes of upwelled water. That seasonal process that I described before also has an episodic influence on krill, driving their density on much shorter timescales of days to weeks. So given this, we would think that blue whales might track ephemeral upwelling plumes within the foraging season and just spoiling it. This is exactly what they do. So using this directional hydrophone that I mentioned, we were able to combine this with in situ and remote sensing of these upwelling plumes that you can see, for example, here, forming at Point Año Nuevo and flowing southward into Monterey Bay. And again, here you see our directional hydrophone on the seafloor. This is really a cartoon version of what was a really data-intensive process, but we were able to show using this instrument that blue whales tend to hang out to the northwest of the directional hydrophone, really sitting right at the location that these upwelling plumes tend to form. When a wind event drives an upwelling event and that upwelling plume starts to drift southward into Monterey Bay, what we observed is blue whales following these upwelling plumes into Monterey Bay and foraging on the really densely aggregated krill that are living within those upwelling plumes. This was an exciting finding just to show another mechanism by which blue whales might be able to more efficiently search this really dynamic ephemeral resource landscape. Importantly, from an applied standpoint, it also told us a little bit what drives the distribution of blue whales in relation to human-wildlife conflict in this system. So really one of the primary threats to this endangered population are ship strikes with large shipping vessels in this system. And it's not really known why blue whales end up in shipping lanes offshore sometimes. And what we were able to show in this paper is that their relative degree of risk of an exposure to those sorts of ship strike interactions is much greater during these periods of time where they're waiting for an upwelling event to occur. And so our hope is that this can play a role in some more dynamic management decisions that I'll talk about at the very end of my talk. One other observation from this work was that we noticed when blue whales were forming these really dense aggregations in upwelling plumes, they tend to produce a different type of vocalization. So this is not the song that I showed before. These sort of vertically downsweeping vocalizations, which we refer to as type D calls, are really rare to record from blue whales, but when we record them, we record a lot of them in a short period of time. And what we noticed is that in these social foraging aggregations of up to 40 blue whales in these upwelling plumes, we were recording a lot of these D calls. So this was an interesting observation, but it really doesn't allow us to test the mechanism of what is the function of this call type and why are these animals selectively choosing to produce this social signal? And so to get at this in a little bit more in depth, we need to identify more periods of time when these anomalous D-call production bouts are occurring. I mentioned Kanoe earlier. She's now working with us on a computer science master's thesis where she's developing an array of machine learning methods to help us detect these really rare events in our long-term passive acoustic recordings. I'm really excited about perspectives Kanoe has brought to this that are sorts of artificial intelligence tools that I was not familiar with before I was lucky enough to work with such an outstanding student. And the sorts of questions that we're getting at using the outputs from those machine learning models are trying to understand really the roles of physical forcing and social or collective behavior in the formation of these ephemeral hotspots of trophic interaction. These are questions like, is the density of the swarm within the upwelling plume, is that a behavioral response to physical forcing in this sort of semi-planktonic animal? Are they forming a denser swarm to hold position in the water column whenever there's a really strong flow that might sweep them out to sea, where they won't have as much access to food or might be more at risk of predation? And then from the predator side of things, what is the function of this type D call in the formation of these social foraging aggregations? There's sort of this paradox here from an evolutionary perspective of what we suggested in the paper where we discovered that they were producing these decalls during the social foraging aggregations is it might be a calling others in to where the prey is. But that's sort of counterintuitive from an evolutionary perspective where we would think, well, that's going to introduce competition here. And so one of the things we're interested in now is thinking about, is this a situation where the food is so abundant and also so ephemeral in its availability that it's valuable actually to call in potential competitors to help you keep track of where that ephemeral food source is? And these are hypotheses. We don't have the answers to these questions yet. But as I'll talk a little bit more about in my Future Visions talk on Monday, really excited about again, integrating with some simulations with our detailed in situ observations that might help us tease apart those hypotheses. I'll just mention one more direction that we're going with this integrated environment prey predator sensing system is to think about these even more cryptic deep diving predators, like things like sperm whales. I have another video here that's probably not going to play, but these sperm whales produce the loudest known biological sounds on Earth. Their echolocation clicks are really incredibly loud signals that we can detect over a great ocean area, again, from a single seafloor hydrophone. Here's a spectrogram we can see here of those clicks that probably will not play for us here, but that's okay. You get an image of the sperm whale as I talk about this. But these sperm whales, it's interesting. We started hearing them a lot in the backgrounds of recordings we've been making in Monterey Bay. And so I started asking folks that work in the whale watch industry in Monterey Bay about this. This is a really large whale watch industry in Monterey Bay, tons of large predators that people want to go see in the ecotourism industry. And I asked people that have worked in that industry for years, are you guys seeing sperm whales? I didn't really realize there were sperm whales around. We're hearing them a lot in our recordings. And what I learned is that people see, maybe a handful of sperm whales over a multi-decade whale watch operation career. And that blew me away because I was like, either I'm getting really lucky of when I listen into when these sperm whales are here, or they're here a lot more than we think they are. And so this prompted a project to develop automated tools to search for these echolocation clicks, which last less than a second in our long-term, 8+ years now, acoustic recording archive. And really the questions that I was interested in here relates to things that we just really don't know about sperm whales in the Northeast Pacific. If you look through the literature on sperm whales in this ocean basin, we know that they move latitudinally over really large areas in this basin, but we really know very little about the context of those movements. We don't know how regular they are, if there's any seasonality to them, and what the behavioral context of them is. This has mostly been from strategic sort of mark recaptures from photographs and some genetics works that's been done. So something I was really interested in is then to leverage acoustic recordings, again, that are being made throughout this ocean basin with automated tools to detect their echolocation clicks in those long-term recordings to try to better understand these latitudinal movements. So for this study, I leveraged two different data sources. One is a long-term oceanographic station in the Gulf of Alaska, Ocean Station Papa, that's had a hydrophone deployed on it at different points in time. And then, of course, also our Monterey Accelerated Research System, that's that seafloor cabled observatory outside of Monterey Bay that I mentioned before. And using these tools, we're able to, again, over many years of recording, construct an average annual cycle of when sperm whales are recorded foraging in proximity to these instruments. And so if we look at the Monterey Accelerated Research System, Monterey Bay area, we see that sperm whales are recorded over the last 8 years in this ecosystem, roughly 50% of days. And this blew my mind when I first saw it, because again, you remember over multi-decadal careers of being on the water every day, people are not seeing these animals hardly at all. So it was really exciting to see that these animals really are very present in this ecosystem. They're just extremely cryptic, but we can take advantage of the sound they're producing to study their behavior. The other thing that I noticed when we did this analysis is that even though these animals are present, roughly half the time, year-round, there appears to be some subtle seasonality in the times of years that we detect these animals, which with a peak more in the late fall and winter and a trough in the summer. Interestingly, this is the exact inverse of what's observed at higher latitudes. So again, animals present year-round, but again with this subtle seasonality, but now inverse with the peak being in the summer. In the interest of time, I'm going to not show the simulations that we ran behind this, but we tested a variety of different hypotheses of what sort of movement behaviors can drive these sorts of patterns in a population over this spatial scale. Movement behaviors that have been previously hypothesized in the literature. And what we found is that the most likely explainer for this sort of pattern is a seasonal latitudinal migration to track subtle resource variability shifts throughout the seasons across latitudes. So keep in mind that these animals are foraging in the deep sea with no direct connection to solar cycles, right? So we wouldn't necessarily expect strong seasonality, but perhaps the seasonality at the surface layer that's then funneling down to the depths as organic matter input comes out of the surface layer into the deep sea might drive more subtle seasonal latitudinal patterns in the deep sea, which is what we think we're recording here. And again, this sort of early observation of these deep sea predators' behaviors is partially about hypothesis generation that, again, are hypotheses that I hope we can test as we continue to understand the data that we've been collecting from this pelagic predator-prey behavioral observatory, which we actually just recovered from the water recently and has now recorded 2 years of data in this system. Okay, so just to wrap up the talk here, I'm going to talk about some work that I've been doing, thinking about how to do community engaged science in this space, both to enhance the efficacy of this research, but also the equity of how this research is conducted. And really this getting into the application of research in this space was again motivated by the extreme dynamism of these systems. It creates a really challenging problem for resource managers to really manage habitats and populations in a system that's constantly moving in space and time. And this has led to the proposal and implementation in many cases of dynamic management strategies where we might think of this in relation to a more static protected area like a national park or something like this, where at any given time step that protected area is in the same place. Under a dynamic management paradigm, this protected area or management tool would be moving in space and time with a population or an ecosystem. And some work I did a few years ago, again, a sort of synthesis review work to look at the proliferation of these dynamic management strategies in marine and terrestrial systems. Each dot on this plot represents a different dynamic management program. And as you'll see here, marine programs in particular really depend on updating the location of this protected area on very fine temporal scales, again, because of the extremely dynamic nature of these ecosystems. And so because of this, dynamic management, especially in oceanic ecosystems, is really dependent on the ability to update management recommendations quickly, often using algorithms and automated data collection. And so in thinking about this, I've been really interested then in thinking about, okay, well, what actually creates these products? What are the data and the tools that actually go into making these products click? And so this is a paper that came out a few weeks ago in Science showing how really dramatically how disparate our biodiversity and behavior data sources are across really what are human dimensions, right? They aren't necessarily representative of what's happening in an ecosystem without human observation, but really may be driven largely by biases in the social human systems that create infrastructures and disparities in terms of who can make observations and what animals or ecosystem processes we observe. So thinking about this in the context of applying algorithms to dynamic management strategies in the ocean or on land for that matter, one question I've been really interested in is how we can enhance the equity of this process given known data disparities and equity issues in data collection, process, algorithmic assessment, and the application of these management tools. So I think there's interventions that can be implemented on really every step of this pipeline, from project development through to how we go about prioritizing where we're going to collect data and on what species and for what problems, really to the assessment of algorithmic solutions, really taking these machine-generated management recommendations and thinking about ways in which we can work with diverse human communities to think about the equity sides of these algorithmic solutions. And so this was a part of a paper from a few years ago, specifically in the context of the open ocean or high seas 30 by 30 agreement, 30% protection by 2030 agreement. But it's also one that applies to more coastal research management applications, things like offshore renewable energy development, for example. And this is something that I've been actively working on off the coast of California. So here I mentioned the Monterey Bay, which is now shown in the north. You're now seeing central and southern California coast, which includes this Morro Bay wind energy area where wind energy development is beginning, which is actually adjacent to the US's first indigenous-led national marine sanctuary, the Chumash Heritage National Marine Sanctuary, which is creating some really complicated and interesting dynamics around how to manage offshore wind development. And to approach these questions, one thing I've been really interested in is, again, using acoustic monitoring to understand animal behavior and the potential impacts of this offshore energy development on animal behavior in these systems. I mentioned we've implemented these stations with the Monterey Bay Aquarium Research Institute. With partners at NOAA, we've implemented several other long-term acoustic monitoring stations. And then most recently, with partners from the Chumash Heritage National Marine Sanctuary and the Northern Chumash Tribal Council, we've actually implemented a couple of more coastally focused monitoring stations focused on shallow water habitats, which are really important for both commercial and recreational fisheries. And this has been an amazing experience to work in this way, in a more community engaged way, and deploying this sort of instrumentation that's often only accessible from a research vessel like we see on the right here. Through Lindsay P.V. Reeves at NOAA, we've been able to form partnerships to go on research cruises with representatives from each of these partners to select the sites and deploy the recorders in this system. Just an example, a couple of quick examples of the sorts of questions we're thinking about using these tools. Ella Kim is a PhD student at Scripps in Southern California, and she's been a partner on this project and has been using the acoustic data we've been collecting to listen to the songs, the choruses of rockfishes, like you see here. And she's been doing a lot of methods development as well as ecological inference using these tools. Of course, offshore wind is coming to Oregon as well. This is something I'm really interested in, not just in the Morro Bay context, but really in the West Coast wide context of how are we thinking about developing this really critical renewable energy infrastructure in a way that's equitable for communities that will be most impacted by their development. One final note while I'm on this topic is that a tool that I'm really excited about for community engaged research are these really low cost, user-friendly acoustic recorders. So I have one here as a prop. This is an audio moth. It's a really low cost recorder that can be deployed both above and below water. And I've been lucky enough to work on a NASA funded project that's been distributing these for community science across the US to understand mostly insect acoustic behavior. But I'm really excited about the ways that this could be used as a tool, both for community engaged science, but also for teaching and really rolling research, teaching, and community engagement all into one. All right, so getting close to time here, just to really wrap things up briefly, really under this whole umbrella, you might have noticed that I'm a huge fan of passive acoustics and bioacoustics. And I think the reason for that that I want to leave you with is this quote from the incredible musician and acoustic ecologist Bernie Krause, who said it better than I ever could, really emphasizing that bioacoustics give us these incredible view into the lives of animals because they contain significant messages for any sentient being. That includes us from both understanding and managing wildlife populations, but also for the wildlife themselves. This is critical social information that allows them to survive, reproduce, communicate, and thrive in wild ecosystems. And while my research program has really thought about that mostly in the oceanic context, I'm really excited about some growing collaborations to hopefully bring this into the terrestrial context as well, to really use bioacoustics as a really key tool for behavioral ecology. So with that, I mentioned my program really is focused on animal behavior and dynamic and changing oceanic ecosystems. My longer term hope is that by really bridging across ecosystems, it really in the long term can be about understanding animal behavior across Earth's ecosystems more generally. This is, of course, work that takes a great deal of collaboration. Thank you to the funding sources that make this possible, also to the incredible artists I've had the chance to work with throughout this journey. So with that, I'll take any questions you might all have. Thanks.
Great, thanks, Will. So we're going to start with questions and consultation. There's going to be tea time and the great team and summer. So you don't get questions answered now. Okay. Awesome. Well, if you don't mind, since they won't be able to hear this mic on Zoom, briefly use it. Yeah.
Thank you. Really fantastic talk. Really enjoyed it. One question. It seems you have multiple hydrophones in different locations that can talk across the coast. How do you try locating the sources of the sound, not just in terms of direction, but actual coordinates through something like triangulation? I can see it's very challenging probably, but I'm curious if you tried and what are the major challenges you cover?
Yeah, it's so the question for folks on Zoom was about getting more spatially explicit with acoustic monitoring via triangulation and not just directionality of the sound. Yeah, this is a huge area of research. A lot of people do this, and it's something that I've worked on a bit. It's a little bit challenging in this broad of a spatial context, just because typically this happens over a smaller spatial scale where you're able to really cleanly distinguish the signal at multiple recording sites simultaneously. This is something that I think is really valuable on a smaller spatial scale and also in a terrestrial context. It's a really valuable tool for understanding movement using acoustic recorders. So it's a huge challenge for that, of course, is time synchronization across instruments, which can be challenging to make sure it's one thing to implement it that way. It's another thing to maintain it when these instruments are in the deep ocean. So that's been a challenge specifically in projects that I've worked on in that area. I will say the one other way that we've gotten at this is to try to really link the passive acoustic recorders with the acoustic recordings on animal as a way to try to understand, okay, so we're hearing a mix of animals from the passive acoustic recorder. Where are some of those animals that we're hearing in relation to one another? And what does that say about what the social interactions might be? And the instruments on animal help us to start getting at that. Yeah. Yeah, it's a good question though. Yeah.
The question I mean, actually, I already answered, but do you think the capacity like default material could be called to other organizations? Not hard to understand like competition.
Yes, so the question was about the D calls from blue whales, which we've observed in the hours leading up to the formations of these social foraging hotspots, really. Yeah, and whether there might be interspecific interactions there. So I think there's two interesting threads maybe there. One is what you suggested about almost like a territoriality of like, okay, blue whales are feeding on this, this is ours. I think the other interesting one here is about the competing pressures that krill face in the ocean, right? So most of the predators that are feeding on them are feeding on one prey item at a time, which might select for very dense swarming behavior, right? This sort of safety in numbers idea. But that's the exact opposite strategy of what a krill should be doing when a blue whale is present, right? Because these are these lunge feeding predators that want the krill to be as densely packed as possible. So one idea that I've wondered about is that sometimes when we are tagging blue whales, we will see smaller cetaceans. So things like Dahl's porpoises, for example, that are really tightly associated with the blue whales and are basically just following them around, picking off bits of krill that are getting through the cracks as these animals filter after they lunge feed. And so I think there's a possibility here, as you might be suggesting, there could be benefit to blue whales of calling in particulate feeding predators to put pressure on the krill to swarm even more. I don't know, this is like starting to get down to multiple layers of hypothetical, but it's the sorts of questions that we're trying to move towards with that observatory. Yeah, it's a really interesting question. Thanks. Oh, yeah. Yeah, so the question was about detecting anthropogenic sounds on the passive acoustic recorders. And yes, absolutely. It's like potentially the dominant noise source that we hear on these instruments, depending on location, it varies. And this is actually one of the pieces we're trying to get at in relation to that offshore wind project I was looking at before, of what is the current status of anthropogenic influence in these ecosystems, just to sort of set a baseline of where do we have more human noise, less human noise, and how might offshore wind construction change that. In terms of impacts on animals, this is not work that I did, but others that we've collaborated with through that long-term acoustic monitoring program have developed automated detectors of what are colloquially referred to as seal bombs. And they're explosives that are mostly used in the squid fishery to deter pinnipeds like sea lions from jumping in the nets to pick the squid out of the nets. And it's not meant to harm the seals or anything. It's really just so you're not catching seals. And one of the interesting dynamics that's come up there is, like, as we there's been some papers published of how frequent and common this practice is and how much of an impact it has on the soundscape. I mean, something I failed to mention is that long-term acoustic monitoring program, you can listen to a live stream of that online on the Monterey Bay Aquarium Research Institute website. And one of the problems that the software engineers that worked on that faced when streaming that audio live was that there's so many of these explosions happening that are really, you know, like startling to the listener, right? So they had to figure out a way to create a little bit of lag, process it, find those, basically mute the audio for a moment so that you don't hear those on the live stream. They're really prevalent in the environment. And one of the things I would be really curious to follow up on in that space is there's information that's come out through conversations with fishermen in that squid fishery about sort of like a dinner bell effect going on here, where like the explosives are meant to deter the sea lions, but in a way they might be calling them in because it's like, well, here's where we're aggregating food for you. And so it could be really interesting, I think, sort of like social ecological project to follow up on that's in this anthropogenic sound influence space. Sorry, I hope I at least in part answered your question there. That was a bit of a tangent, but yeah.
Tea time at 2. I fucked up the